Welcome, everyone. Welcome as you get settled. We'll be starting the webinar shortly. So I see there's still people joining. I'm going to give it about 30 more seconds and then we'll get started. Well, wonderful. So good morning, good afternoon, or good evening, depending on where you are in the world. And thank you for joining us today for the Multi-Regional Clinical Trial Center of Brigham and Women's Hospital and Harvard webinar entitled Returning Individual Research Results and Data to Participants, Experience from the Field. I'm Sylvia Baydorf-Casas. I'm a program director here at the MRCT Center, and I have the pleasure of introducing the topic today and moderating the discussion that we'll be having in a little bit. Before we get started, I have a few housekeeping items that I will go over very quickly. One is that we're recording this session, as you can see, and the slides and the recording will be available after this webinar within the next few days on the MRCT Center website, so stay tuned for that. There's also closed captioning available for this webinar, so please feel free to go to the Zoom toolbar and enable closed captioning for you to be able to see the transcript of what I'm saying and what our panelists are saying. Third, we have the Q&A function enabled. So if you have any questions throughout this session, please don't hesitate to put them into the Q&A. We would prefer the Q&A over the chat, please. So please use the Q&A because that's a way that we're able to track the questions and responses more easily. And our panelists can also type responses where applicable as well. In addition, a new thing that we're trying here at the MRCT Center is to add a little survey at the end of the webinar. So when you close out of Zoom, you'll see a survey pop up with 3 questions that will ask you a little bit about your experience with this webinar and the subject matter. So please, if you'd be so kind to volunteer for that anonymous survey and give us your feedback, we would appreciate it. And lastly, before we get into all the content today, the opinions expressed today are those of the speakers and not intended to represent the opinions or position of Brigham and Women's Hospital, Harvard, or any other organization. The MRCT Center is supported by voluntary contributions from a variety of different sources, but regardless of who funds us, we are committed to the autonomy in our work and to transparency in our relationships. There are no conflicts of interest related to this presentation to disclose. So for those of you who are not familiar with the Multi-Regional Clinical Trial Center, we are a research and policy center based out of Boston and Cambridge, Massachusetts in the Northeast of the United States. And our vision is to improve the integrity, safety, and rigor of global clinical trials. And this has been our vision since we were established about 13 years ago by Dr. Barbara Buer and attorney Mark Barnes. Our mission is to engage diverse stakeholders to identify and define emerging issues in the global clinical trial ecosystem, and together create and implement ethical, actionable, and practical solutions. And today you're going to hear a little bit about the action that some of your colleagues have taken in this area of individual research results return. The MRCT Center does its work by developing standards, identifying opportunities for improvement, establishing best practices, and improving transparency. And that last point is really important in the context of this conversation, because transparency and trust is what we're really trying to build in this work of returning individual research results to participants. The work that we've been doing in this area has been ongoing for several years now. Back in 2015, we established a work group to create standard guidelines and criteria related to returning individual research results to participants. That work resulted in a toolkit and guidance documents that were released in 2017, and they were out in the world being used by many of you and also not being used by many of you. We learned over time that there were many operational and just challenges to implementing individual return of results. And so we decided to revisit, and this is something we do with many of our projects. After several years of them being out in the world, we take a look to see if there's anything that needs to be refreshed, updated, changed. And so in January 2021, under the adept project management of Lisa Murray, there was a task force that was established to create updated operational and implementation-oriented tools and resources. These were released last year in March of 2022 in a project-specific website. And then over the last year, we've been working on a few case studies to share with you, which were released just last month in April of 2023. And here we are today to discuss them. Just as a refresher and reminder, this is the Return of Individual Research Results website. You'll be able to find the case studies under the Resources tab. But I also just want to quickly bring your attention to the Getting Started section and How to Return IRR, because these are some resources and tools that will be extremely helpful to you if you're thinking about getting started with returning results, or have been doing it and running into challenges and may want to see some tools and resources that could help you. And these are definitely going to build onto a lot of the things we're going to talk about today in our session with our panelists. So these case studies span the gamut from talking about a participant's experience of receiving results and a decision-making tool that was developed around those types of results being returned, to routine lab results being returned to participants, to some of the trickier issues that we often think about around non-validated test results and responsibly returning secondary findings. I will note that our second case study here on returning routine lab results, which was authored by Megan McBride at Janssen, won't be featured today because she sends her regrets and is unable to be here today. But nonetheless, that case is available on our website for you to review and to learn more about how Janssen tackled returning results in their studies. So then, without further ado, I'll just bring us to the next phase of our webinar today, which is each of our case studies will be introduced by our panelists. They'll introduce themselves and they'll tell you some high-level details about that case, what they did and what they learned. We'll go through each of those case studies that we have. We have 4 case studies to discuss today. And then we'll go into a discussion, a panel discussion, where we'll have some questions and answers that we have for each other, and then also take questions from the audience. So with that, I invite you, Jamie Tyrone, our patient advocate, and Doris Allen from Virginia Tech, to tell us a little bit about yourselves and supporting participant decision-making in genetic testing studies.
Greetings, this is Jamie.
Jamie, come on camera too. Come, you're off mute. Come off mute and join us. You did it. Welcome. No problem. Take a deep breath. And here
I'm
we
a patient advocate and not a technologist. So anyway, thank you so much for including me in this very, very important subject that is near and dear to my heart. And I would like to share with you my experience. I'm a patient advocate. I am a research participant for 13 years for a longitudinal biomarker study for Alzheimer's disease. I have 2 copies of a gene that put me at a 91% lifetime risk. How I found out about this information is I participated in a study. I was having neurological issues, and I participated in a study that was done mainly like a direct-to-consumer product, that if you knew your status for certain genes, would you change your lifestyle to prevent disease? I was interested in my genetic status for MS. even though I had a very, very strong family history for Alzheimer's disease, but I didn't equate the two. So I eagerly signed up for the study, signed an informed consent that was all done online, just like when you download an app that you want so badly, and didn't read the fine print, and discovered one evening by myself, without any genetic counseling, any support, any information that was given to me, that I had 2 copies of the ApoE4 gene/allele that put me at this 91% lifetime risk that I found out that night by Dr. Google. And it threw me into a tailspin. My father was dying with Alzheimer's disease, and I didn't know what to do with this information. I subsequently went into a very deep depression. I was diagnosed with post-traumatic syndrome. I am embarrassed to say, but I do share it because it's an integral part of my story, that I did contemplate ending my life. I had a plan, and I knew that I needed some help. In the meantime, I discovered a book written by Dr. Doris Salin. And the book's titled Test or Not to Test. And I knew at that moment that what happened to me should never happen to anyone else. I was validated. The PI said I should be happy to know this information because I can change my lifestyle to prevent disease. There's no prevention or cure. There is just decreasing your risk. So Dr. Doris Salin's book became very instrumental in the beginning of my healing process. And I'd like to introduce you to Dr. Salin, and she can share with you a little bit about her book and her experience.
Hello, thank you very much. I'm a professor emerita of Science Studies and Humanities at Virginia Tech. And the book that Jamie's referring to examines genetic testing by drawing on the experiences of over 200 people who had genetic testing for a variety of medical disorders. Some of them had genetic testing, and many others decided against it. The main finding from the book is that preparation is important before having genetic testing. And as part of this preparation, several questions should be considered. One of the key questions is, does the value of having the genetic information, the benefit from the information, outweigh the possible burdens that the information can bring? When we learned of Jamie's experience, we felt that we had to convert some of the findings of this book into an educational tool and a decision aid. And this decision aid would be directed toward helping people make better informed decisions about their genetic testing for their risk for Alzheimer's disease. We've developed a prototype while doing it, convened several focus groups. We consulted with genetic professionals and physicians. And once we had the prototype, we tested it further in a rather large study involving 1,200 participants who gave us feedback on the tool. And the result was 93% of them found it helpful or very helpful and would recommend it to others. Of course, they also had some suggestions for improvements, which we made. So you can check this out. The link is on our case study, and it's freely available online.
And I would like to punctuate the fact that I did not know that Doris was creating this tool until it was complete. It's about a 2.5-year process. And when I used the tool and put myself at that point in time in which I received my results, it came back that I was not prepared and I was not ready. And so this tool has become near and dear to my heart because it would have prevented me from going through the pain, the cost of psychological care, the trauma that I endured. And now that I have a few moments, I'd like to take this opportunity. Doris doesn't know about this, no one does. It became clinically significant. I had an MRI that showed that my hippocampal volume was 32% of what it should be in the clinical setting. My neurologist wanted to compare it with the images that I had for the previous 12 years in this longitudinal biomarker study. I was denied the results because the study was not designed that way. And so I am happy to say that I am meeting with them next week, and my lab rat duties are in Arizona, and I will be getting those results, and they will be disclosed and given to my neurologist. And I just want to say how much meaning there is in giving these results to participants, and what it can do to empower our lives into helping make us decisions for us. Thank you.
Thank you so much, Jamie. And it is such a key issue because, you know, we're going to talk next about what Pfizer has been doing, and Paula Boyles will be introducing that case study. But the idea of being able to get those results that could have some clinical significance moving forward is really key. And so it's just wonderful that you've been able to share this and that you'll have the experience and opportunity to get those results next week. Thank you, Jamie.
Thank you.
Paula Boyles, you're up next. Please tell us a little bit about your case study and yourself.
Thank you, Sylvia. So my name is Paula Boyles, and I'm the lead for external clinical trial data sharing at Pfizer. And one of the initiatives that we've been focused on is participant data return, which is essentially returning an individual's clinical trial data back to them at a specified time. And I'm pleased to say that beginning in June this year, Pfizer will start returning data back to individual participants for all studies with US sites at the end of the study. And so the case study that we have presented is how we have managed to achieve this. And so starting off, we had the planning phase, and we were really looking to get organizational buy-in from top levels of the organization all the way down, and also across divisions, because really the impact of returning data reaches across so many different organizational lines. So next came our market research, which was very key. This was to inform and shape what the data return solution might look like. And we were looking to answer questions such as, what data do you want to see? How do you want to see it? When would you like the data? What format would be most useful? And so once we knew all that, we then moved into the build phase. Now, build was really broken down into 3 interdependent components. We had the creation of the data file itself. We had the data return infrastructure, the technology piece. And last and certainly not least, we had the participant experience and the end user access. And so our solution was really designed primarily to use current infrastructure where possible. And we're looking to pull our data from our clinical data analysis and reporting system and populate a PDF template. Now, the PDF report will be stored in a very secure area, which has been custom designed. And then it will be shared with our participants via our Pfizer clinical trial alumni portal. And participants are invited to create an account on the portal, and they basically opt in to receive their data. And most importantly, this solution ensures that the data is returned in a secure manner, protecting an individual's privacy at all times. That's very key. Now, building a solution is one thing, but change management, ensuring that the organization is ready to adopt a new solution, is really crucial to the project's success. And in my opinion, it's probably one of the most challenging aspects of this project. For participant data return, it's essential that it's considered early on in the study planning phase. And so we're introducing a data return plan, which will be completed at the clinical development planning stage, right at the beginning of the study. And that'll be used to define special study-specific data needs. And we're also updating some working practices so that the creation and the QC of the data return package becomes just a standard part of our end of study clinical trial reporting process. And I should point out that all of this requires resources, it requires funding, and it requires time, all of which are very challenging to achieve, especially when people have already full-time day jobs. So it's essential that significant planning takes place, and that's a necessity to ensure successful implementation. So is this what the final solution looks like for Pfizer? Absolutely not. Moving forward, we will be looking at returning additional data types, such as imaging files and potentially genomic data. We'll be looking at returning data for EU studies and rest of world. We'll be investigating the possibility of returning data during the study, and also looking at alternative ways of returning the data, such as integrating into electronic health records and similar tools. So there's a lot more to come. And to conclude, returning data for Pfizer studies, that's great, but our hope is that our data return solution can act as a springboard for others in industry to learn and begin creating their own data return solutions. We actively work with other industry consortia such as Transcelerate, IMI Facilitate initiatives, and we try to influence and help develop guidance for participant data return. And I look forward to a time when returning participant data is just the norm across industry worldwide. So that's where Pfizer is at.
That's wonderful, Paula. Thank you so much for sharing that. And really the idea that this is inspiration, this is fodder for others to be able to follow that model. We appreciate it.
Thank you.
We now have Linda Coleman from Yale University, who's going to share a little bit about their experience returning non-validated test results during a global pandemic, nonetheless. Linda, welcome.
Well, thank you so much. So my name is Linda Coleman, and I am the director of the Human Research Protection Program at Yale University. And as was mentioned already, what I'm going to be talking about a bit is how our IRB navigated returning results based on a new COVID-19 assay that was being developed, which is from a non-CLIA certified lab during a public health emergency. And so, of course, the case studies have a lot more detail. I won't go into some of the regulatory issues involving what kind of drove or what prompted the work that we did, but it is referenced in the case study. So here's the basic background. So a researcher was creating a repository for current and future research involving the epidemiology and pathogenesis of emerging viral infections, including the coronavirus. And one of the secondary aims was to validate a university-developed assay to test for COVID-19, which was, obviously very much needed. These samples were being collected from hospital inpatients and outpatients who were either suspected of exposure to COVID-19 or having symptoms. And in this particular case, samples were first tested in a non-CLIA certified lab using a university developed assay, this assay that we're talking about. And then the second piece to this was that these same samples would be retested in a CLIA certified lab to validate the results. As some of you may recall, especially early on in the pandemic, there was a significant backlog in terms of trying to get test results returned from labs in general, not just COVID-19 test results, but all kinds of labs. There was such a backlog. And because of this, the IRB really needed to work with the investigator or the researcher and the HRPP to figure out what should we do in this case. In a situation where this, non-CLIA certified test results come back and potentially a test is positive or could be negative, but is potentially, especially positive, what should be done? Especially in light of the fact that there are particular regulatory requirements that need to be followed, and namely there's the CLIA regulations, and there are also others as well. So because of this, a decision was made that because of the unique circumstances of this public health emergency and the potential contagiousness of the situation, the fact that you're putting not only providers at risk, you're putting potentially non-subjects at risk, the decision was made that there was an ethical obligation to alert providers, the healthcare providers of these particular research participants, of a potential unconfirmed finding of a positive COVID-19 test. And a lot of work went into how to communicate that. So for example, so in this case, the decision was made to verbally communicate this information to the healthcare provider so that that provider could then take the appropriate clinical action. There was the IRB wanted to ensure that these samples were retested in a CLIA certified lab immediately, and that those CLIA certified results will be returned directly to the provider for any additional required action, including any sort of mandatory reporting to health authorities and hospital infection control authorities as well. So some other things that the IRB spent a lot of time discussing was how to communicate this to participants themselves, operationally, how this would work. And again, really kind of focusing again on, how do we ensure that we adhere to regulatory requirements while at the same time, balancing the potential risk to the participants, to the providers, and to non-subjects, and also ensuring scientific integrity of the study as well. And because the idea being that we would want to make sure that these lab results were retested. So that is really the case study that we will talk a little bit more. There'll be some additional questions on this, and I can spend a little more time on this, but that was really the focus. How to navigate the regulatory requirements and the ethical obligations during a public health emergency.
Thank you so much, Linda, for sharing your experience at Yale. And last but certainly not least, we have Megan Frone from the National Cancer Institute, and she's going to talk a little bit about responsibly returning secondary findings.
Yes,
Megan,
hi.
Welcome.
Thank you. So hi, everyone. My name is Megan Frone, and I'm a board certified genetic counselor in the clinical genetics branch at the Division of Cancer Epidemiology and Genetics at the National Cancer Institute at NIH, because the government really does love its big titles. And in my role at NCI, I provide clinical genetic counseling and testing and return of results for several of our clinical natural history protocols. And I'm also involved in writing the genomics portions of our protocols, which includes return of results. And then my other primary role is genetic variant bioinformatics, annotation, curation, and reporting for various efforts throughout the division. And so through my various job responsibilities, I'm really involved in return of results at every point in the process, from when we first propose a project and begin to write it up, to encountering reportable variants in the analyses phase, to thinking about how to report them and craft those reports, and then to returning them to participants and patients and kind of all the downstream of that. So I was honored to be approached by the MRCT to help in the individual return of results initiative with a focus on secondary findings. So we worked together to create a hypothetical case that was grounded in real world examples that we pulled from various challenges that teams are often encountering when they're designing and implementing research studies that involve genomic analyses. And we really wanted to highlight the main aspects to consider in return of genomic findings in the research setting. And the real takeaway that I hope that people come away with from our case is that return of results really needs to be consideration at every point in the planning and developing and implementing of genomic research studies, because it's not if secondary findings will be encountered, but really when. And through having thought through how a study team will address secondary findings and how this will be communicated to study participants, and thinking about that at every step in the process, not only will that make things much easier, but it really does ensure optimal care and experience for the research participants. And it really is just best practice for genomics science at this point. And it's far easier and more straightforward to implement than I think we sometimes think it is. So I'm happy to be here today to talk a little bit more about the case and answer questions. And I'll also just note that as a government employee, my content and views today are my own, and they don't necessarily reflect any official positions of NIH or HHS. So thank you for having me today.
Thank you so much, Megan. So we will get into the discussion now. So what I'll do is invite everybody, I'll stop screen sharing, and I'll invite everybody to come on to start their videos and come on camera. Okay. So we'll be able to have a little bit of a discussion. I want to start, Jamie. I'm bringing you, I'm calling you, I'm invoking you to come.
For some reason I can't get my video to go. Start my video. Okay, there we go.
Hey, well, Jamie, I wanted to start with you because so much of what we're doing here is really for participants and patients, right? And you've described this really personally difficult experience of returning really difficult results. And yet you're still supportive of this. You're still a champion for this. You're an advocate for this. So I always find it really helpful to hear from patients with this experience to tell us, what can sponsors and researchers do to prepare participants to understand and to receive the results, to understand the meaning of those results? What can researchers and sponsors do, from your opinion?
Well, first, what I would like to say is that I've become an advocate for results because they are very, very important. And unfortunately, or fortunately, depending on how one looks at it, is that I've experienced both sides of it, how not to do it and how to do it. And I think education, counseling, preparation for what these results might mean to somebody so they can actually decide whether or not they're ready to know these results. I was not ready to know these results in the beginning, but as my journey has progressed, I realized those results can be very empowering. I do believe that at some point in time, it's going to be very important to get the research participant involved in study designs and what it is that we need and we want, that it needs to be done correctly and safely, and that it is so empowering to be a part of the process. And so I now, as a research participant for the longitudinal study that I've been in, they consult me, they involve me. When I asked for my results, they kept saying, we want to get this right. We want to make sure that it's safe. We want to make sure that you're prepared. And so I think really listening to the research participant and getting them involved. And instead, hopefully one of these days, the nomenclature will change from research to participant to research partner, research collaborator, and to be a part of the process. And Paula, as I heard and listened to your presentation, I'm just so excited to see how far we've come in this process. I've been doing this for 13, 14 years. And people were looking at me like I had 3 eyes, like, oh, this is never going to happen. But guess what? We're there. And I get asked all the time, well, I don't know how we're going to do this. This seems like an awful lot of work. I'm like, no, you don't understand. MRCT's done a lot of this work for you. And so it's something that I now have in my toolkit as I talk about results and returning it to patients. That's just such important work that the MRCT has done, and to have allowed me to be a part of the process. So I just want to thank you for the empowerment that I've received. And one of the things that I would like to say, and Barbara has heard me say this last week, is that when I was in nursing school way back in the day, I was told that medicine is not necessarily about curing, but it's about healing, because we're not getting out of here alive. And letting the patient, empowering them, is part of the healing process. And every one of you here has the opportunity to allow us to heal as we go through our life's journey in being a research participant and trying to make a difference.
Thank you, Jamie. And I mean, this idea of healing is such a huge one, because so much of the work we do, we feel like we're rushing through it. And, you know, this gives us a moment in our busy schedules to take a moment and pause. And so much of the theme of today is the preparation, the planning, the thinking through what kinds of results might be produced in the study, and when or what can be returned and how to return it. So the idea of thinking through it and really doing that thoughtful exercise, which we do have tools for, on just selecting what to return and how, is really key. And I think your partner in this, Doris, not every study is going to have a robust decision-making tool to help participants or potential participants make decisions around participation or receiving results or things like that. But I imagine that in this process of creating a decision-making tool, that you've had some more general learnings or considerations that other researchers or others who are looking to help participants make decisions around these important issues might be able to glean from. So do you have any little pearls of wisdom to share about your experience that other researchers might benefit from?
Well, how much time do we have?
You have lots of pearls. This is great.
Of course, it's not practical to follow the same lengthy process that we did. It took us over 2.5 years, although some of that was time spent trying to find funding for this. But we learned a lot from our study, especially from all the feedback we got from over 1,200 people. And there are some pearls of wisdom that would help speed up the development process for others who wanted to provide such materials. And the first thing is, people want simplicity and clarity. They don't want to be confronted with enormous amounts of material to read, large amounts of data. You can include extra links to to more detailed information for those people who want it, but nobody wants to be confronted with that at first sight. They want the value components, the benefits, in this case, the benefits of genetic information, as well as the burdens of genetic information, to be presented in a balanced manner. And they want the tool to be responsive to their learning styles. There are people who are comfortable with text. There are people who just want bullet points. And there are the visual learners who want some diagrams or figures that can help them better understand statistical data or risk estimates. So you have to be prepared to reach out to people who will want to learn in different ways. It's important, though, always to keep in mind that tools like this, such as we developed, are not meant to replace the detailed discussions with the study team. They're meant to provide some background and enhance those discussions when they take place. Of course, you do have to test them before you use them widely. It could be done with a small group, but you should be looking for whether there are any glitches in especially the online tool, and whether it addresses all of the major concerns that people would have.
And that's key. I think that involving, this is what Jamie was saying too, involving stakeholders. And Paula, you were saying that too, in terms of some of the testing you had to do to make sure that it's being delivered in ways that can be taken up by the recipient.
I do want to add that there's always an undercurrent that maybe if we assault people with too much information, it will deter them from participating in the study. But here's what we found when among our 1,200 or so participants in our study, about just over half, maybe 55%, decided to change their minds about genetic testing. And 70% of those who changed their minds decided they were more willing to have genetic testing and have genetic information. So giving people background help with thinking through the kinds of information they want doesn't seem to deter them from participation in research.
Which is a key question that researchers will have and sponsors will have, is that too much information going to keep people from joining the study? But we always lean towards the idea that more information is better for people to make informed decisions and then stay in the study because the study's meeting the expectations they had. Well, so Paula, I have a question for you. There's a few questions for you in the Q&A as well. And I mean, the main thing is that most industry-sponsored studies are not returning results right now, right? That's not necessarily the reality. So why did you decide to take this on at Pfizer, and why did your team do this? And did you get any pushback or experience resistance in that process of starting this work?
Okay. Well, we heard from Jamie that, you know, patients, patient advocates are crying out. We're hearing loud and clear that they want their data. So, you know, that's a message that we have heard loud and clear. And as an organization, we're committed to health equity. And, you know, we feel that it's important that we want to listen and build trust with our participants. And so, you know, at the end of the day, trust is everything. And by returning the individual data, we want to demonstrate that we hear you, you know, we're committed to doing what we can to meet your needs and improve your overall trial experience. And ultimately, you can trust us to do the right thing. And quite honestly, returning data back to participants is just the right thing to do. So it was really, that was the driver behind it all. It's just the right thing to do.
Well, and a couple of people in the of our participants are asking, so how soon are you returning individual results? Is it, you know, so how soon are you sharing it? And is this be done for patients in the US or globally to all? There's a few questions I'm seeing coming up here.
Yeah. So we will begin returning data in June. And we will be returning data for all studies moving forward, all studies with a US site, and data will be returned at the end of the study. So it will become a standard practice for all Pfizer studies moving forward.
Yeah. And how soon after a study is completed?
I'm sorry?
How soon after a study is completed are those individual results shared?
So after the primary completion, it will be 12 months. So it's actually in alignment with when we currently report our aggregate results of the study, our plain language summary, which is the the plain language version of our study results. So moving forward, participants will be able to see not only the aggregate data of the overall study they participated in, but their own individual data which contributed to that study.
Perfect. Thank you, Paula, for clarifying that. So I think we want to hear from Linda too. And I see there's some questions here that are kind of CLIA related from Jim Feldman. Hi, Jim, welcome. So you had to navigate the regulatory landscape, the ethical landscape. You had to bring you had to cross into a lot of different places. The IRB doesn't necessarily always cross the HRPP
Right.
in terms of clinical care and navigating sort of the Department of Medicine, I believe, or others that you had to think through.
Right.
Can you tell us a little bit about that? And also, while you're doing that, Dr. Feldman has a very specific question around non-CLIA certified results.
Right. I saw that.
So great. So if you're able to kind of comment on that and
Sure.
that experience, thanks.
So very complicated situation because as some of you may know, if you look at some of the HIPAA requirements and CLIA requirements, they're not actually harmonized in terms of, you know, some of the requirements in terms of when you're returning results in a situation where someone is going to be using those results for the diagnosis, prevention, or treatment. I think that's the key. If you're doing, if you have research results and you're not using for that purpose, of course, you can return, you can share that information, but for purposes of the treatment and prevention of disease, et cetera, for what we were talking about a pandemic, somebody who might have suspected COVID-19 and, you know, COVID-19, that kind of thing. We really had to figure out how do we manage that. In today's environment, it wouldn't be an issue, right? Because we would have this university developed assay. We would not return those results. We would be immediately sending it to a CLIA certified laboratory, and we would get those results returned directly to the provider, which then would be documented in your, you know, EMR or however you document it, and it would be shared, and then their clinical action could occur in terms of the provider. And also, you would also have some additional obligations in terms of reporting obligations as well, right? Because of the fact that this is a you have to report to other health authorities as well. So in this case, the central issue is the fact that because of the delay, we had to figure out a unique way of providing information without violating CLIA, right? As a precautionary measure. So maybe I wasn't clear about how this worked. We verbally had the researcher contact the provider. We had a process in place where it was documented that the person actually had a communication with the provider. Like, it wasn't just a voicemail or an email. It was like, I just want to let you know that we have this university developed test. It's not, you know, CLIA, you know, it's not been done in a CLIA certified lab, but we think you should know because who knows when we're going to get the results from the CLIA certified lab. So I understand, you know, the question you had about the documentation. That was what took so much time. So the IRB, the investigator, me as the HRPP director, the hospital, we had to figure out how do we navigate this, because normally you would just put this into the record, right? Your electronic health record, and it would all be there. But because of the regulatory issues, we didn't feel like we could do that, but we still felt like from an ethical perspective and from a clinical perspective, we needed to share this information. So that is kind of how we kind of got around some of these regulatory issues. And as I mentioned, in today's environment, we would need to do that. But in the environment in 2020, we needed to figure out a way to do this, to protect the providers, to protect the participants, and protect third parties because of this risk. I'm not sure if I answered the question, but you know, that's really what we were trying to navigate.
Thank you, Linda. And I think I remember having been at the IRB myself, there was always this question around the CLIA notification and validation and making sure, but this seemed like it was a very special circumstance that you could be able to, and keep the letter of the law, stick to the letter of the law while
technically, yes.
Technically,
but do the right thing,
which is, I think, something that came up in our discussion, right? That sometimes, you know, you really have to stretch to find that way to stick to the law, but also do what was right. So thank you. And so Megan, a question for you, because secondary findings, as you've said, they're going to come up in genetic and genomic studies. And the idea that we have a lot of planning and preparation we can do in advance. Your case study really outlines like when there's been planning and prep, how people can follow the plan and bring bring the return of results to fruition, and also help a patient with a potential clinical care issue. So do you want to share a little bit more, just like how can researchers really prepare and plan? Like how do you recommend that people do it? Like, what's the first step they should take? How do they do it for their studies?
Yeah, I think, again, and I'm going to sound like a little bit of a broken record with it, but the plan should be made up front. And that should be described in the study protocol and communicated to participants, right? I think we're hearing kind of a common thread throughout all of the different speakers here about when and how they'll get the results to be returned. And this is not a one-size-fits-all process, right? Every study, every institute is going to have a very different plan. But what is your plan going to be, right? Is it going to be electronic return of results? Are they going to get a call from study personnel? Is their primary care doctor going to get the results and communicate it to them? Is it going to need to be CLIA confirmed before they get all of their results details? How are they going to understand what the results mean and what they mean for them? Who can they contact if they have questions? So this needs to be clearly communicated to the participants, and the researchers need to know that in order to be clearly communicating that. And they should be thinking through the logistics and the study resources they have for return of results, because depending on the type of results they're planning on returning and how they're planning on doing it, that can be a pretty big lift. So one of the things that, the key things that we recommend kind of early in the process is think about how many study participants that you estimate will have a finding that's going to be returnable, because that may help you think through scale and scope for which of these methods of return is going to be realistic and the most effective. I think the other thing too is I think sometimes or researchers are thinking about the time frame, right? And that's also going to vary significantly by study. There's not necessarily a requirement to get these secondary research results into participant hands on any sort of timeline, especially if it's not the primary aim of the research. And that's fine, right? But it should be made really clear to participants up front in the protocol and during their consenting, what secondary findings may be returned and when they may be able to expect that. And it may be that there is no timeline, right? Especially if there's no given timeline for even when the sample may be analyzed, right? Some patients are biobanking, but we may never requisition those samples to look at them. So participants should understand that, right? And if they, and that also is important to understand that if they haven't been contacted with the results, it doesn't necessarily mean that their testing was normal or negative. It may not have even been done. And even when it is done, not everything's going to be looked at, right? Because that's not necessarily the aim of the study. So, if they have health concerns that warrant genetic risk assessment outside of that, that should be done clinically with their healthcare providers kind of outside of the research study. So just all of these different aspects and kind of thinking about them beforehand is going to save you a lot of time on the back end when they inevitably come up.
Thank you, Megan. And you also mentioned something. So when you think about, you're doing this on one site, but what if we're doing it on multi-center studies? There's a couple of questions coming up from an operational standpoint. And so Paula, you might be able to answer this, Linda too, thinking about, from a multi-center perspective, how does dissemination get handled? And I think there's some questions here about the best way of sharing individual results. I mean, we're talking about a portal that people opt into access. Are there other ways that the people are familiar with or have experience with?
Jamie has her hand up. Go ahead, Jamie.
So with our agreed group, we just published in the Journal of Alzheimer's Disease a proposed patient's Bill of Rights, participant's Bill of Rights. And in that Bill of Rights, what we do is we propose that the patient decides on how they want to have these results done, given to them, whether it's writing, whether it's verbal, who they want to be there, a family member, no family member, because if it's genetic, it may affect the family. Some family members don't want to know, others do. But if as you're as you're designing these studies, if you can kind of keep that in the back of your mind, that let the patient decide how it is that they, if it's feasible, how to receive these results.
At least try to take that into account. Linda,
Of
were you, or
course.
Doris, were you going to say
I
something?
think sometimes they may not want the results or may not want it at that time, and that has to be respected.
Right. And as I mentioned, you know, at Pfizer, when we were planning how to do this, we reached out to listen and try and understand what it was that the patients themselves wanted. You know, and we heard that, you know, as much data as possible is what was required. And you know, that there was definitely preferences for having you know, a simple PDF format for the document that would make it easy to reuse, to share with you know, the primary care physician or upload to their electronic health records. So yeah, certainly the solution that we came up with was not something we devised ourselves internally. The solution was really created and designed by past study participants, healthcare providers, and patient advocates. It was all based on their input. It's true.
What about the situation, genetic data, in which people do not want the data in their medical record? Is there a way to handle that?
So currently, for Pfizer studies, we are not planning on returning genetic data at this time. I won't say it's never going to happen. It's certainly something we have in scope, and that's, you know, that's a very good question that we'll be looking at in the next phase of our delivery.
But Paula, it doesn't go directly into the medical record, does it?
No, no.
So
The data will be returned into the alumni portal, and then it's up to the individual to decide what they want to do with it. You know, if they want to share it, upload it to electronic health records, that's entirely up to them. It's their data to share how they want to.
Jamie, do you have one thing you want to add before we start wrapping up? Please.
So one of the considerations that I fell into was that once I knew these genetic results, I am now open to discrimination for different types of insurance policies, life insurance, long-term care insurance, disability insurance. So that, I think, is important to disclose to the patient before they're even offered the return of results, in case they want to get their financial house in order, certain other considerations they may be taking, almost as part of an informed consent. I mean, as an informed consent, really, truly, that they've been informed that this information could be used against them from a discriminatory perspective. Some may still want to know, some may well say, okay, hey, let me put some things on hold, let me get this stuff in order. But I think they need to be offered that opportunity and that knowledge.
And I do know there are a number of questions still in the Q&A that we haven't been able to get to. We'll do our best to respond to some of those in writing and put them as a resource with the recording and slides in a few days. So I want to pass the baton to Barbara Beer, as I mentioned, our faculty director, who will conclude with a few thoughts and send us on our way. Thank you to our panelists.
So first of all, thank you so much for this very, very rich discussion. And I can't tell you how many questions have been answered, posed, and continue to come through, which just sort of suggests how the more we focus on this, the more, you know, there are specific questions that we really need to figure out and have a cooperative way of doing it. And consider each of these as individual case studies for the time being until we can figure out sort of whether there are system solutions to make sure that participants have a choice, that they're well informed, that they understand it, and that the data is available to them if they can have it. We've touched on what I think is the second, as it were, easiest thing to, or third. I think it's the easiest to return in terms of complexity is the aggregate return of results. But everybody who receives those results will say, what about me? The second would be to say, what arm are they on? And the third is sort of clinical results, clinically validated results, but for the participation in a research study would be the same if they were drawn in a clinical setting. Very different than true research results, where I think the questions of what is the utility and what can we do with it and how can we share it, even if it's not intended for health decisions, is really complicated. And yet we have to find our path forward. I think that what you all have done has been really brilliant, moving this area forward in a patient-centric, really thoughtful way. And we look forward to staying in touch, bringing more case studies together and forward, and also, sort of thinking through those issues that we failed to address before that we should continue to think about together. So folks on the call or who hear this, who have ideas, please do bring forward your case studies and bring forward your questions, because we have a very committed group of folks that are really in the trenches of trying to do the right thing. And with that, let me thank you all again. This was a wonderful discussion and really illuminating for me, and I'm sure for the audience. Thank you.
Thank you.
Thank you all.
And we're seeing little floating hands come up from the metaverse,
Little hearts, little trumpets. We appreciate everyone for attending today.
Thank you.
Appreciate our panelists so much.
Thank you for
having us.
thank
Thank
you
you.
very much.
Very much.
Many great questions. I know we're still live. So many more to think about. Kristen, are we able to stop broadcasting and
Not
recording?
usually. We can stop recording.